بسم الله الرحمن الرحيم. السلام عليكم ورحمة الله تعالى وبركاته. How are you, dear students? I hope everybody is doing very well and in very good health, inshallah. Okay, so this is the video for our lesson 5. So in this lesson here we are going to talk about little gene and 6 linked inheritance. So I will move to the slide. So little gene, this one I already explained in live session, but never mind, since some of you they requested for the video, I'm making the video for them. So actually this one started when some researchers started in 1904. They started to cross black mice with yellow mice, and so they started to do this crossing, so we knowing that the yellow is dominant over the black. So the black mice here is actually true breeding, and the yellow mice, so when they do this kind of crossing, what they get, so here I prepared the Punnett square. When you cross a black with yellow, let's imagine that the yellow one is actually true breeding. So what are you going to have? So since the yellow is dominant over the black, we are going to annotate this one capital Y and the black will be small y. So here the yellow one is the gametes are capital Y, the black will be small y, small y. So the offspring will be capital Y, small y, capital Y, small y everywhere. Means the offspring will be all yellow, 100% of them yellow. So this is what we can predict, right? If both parents are true breeding. But when the researchers start doing this a kind of experiment, they can never get actually this ratio. They never get this ratio. So what they get is, when they do this kind of experiment, they got always 50% black and 50% of the mice are yellow. So this one, what does it suggest? It suggests that let me silence my phone. It suggests that the yellow is actually a heterozygote. I had to silence my phone, sorry. Okay, so it suggests that yellow mice must have been heterozygote. Okay, so means it will be capital Y, small y for the yellow and small y for the black. Then your offspring will be capital Y, small y, capital Y, small y, and here small y, small y. So this one are 50% yellow and this one are 50% black. Okay, so this is the first part of the experiment. So this is your F1. Now what they are going to do, they are going to cross the yellow one with yellow one. Okay, so if we cross the yellow one, F1 with F1, but we cross the yellow ones, so by right this one we will have capital Y, small y on here, and here capital small y. Then we will have this combination. Means this combination is, we can predict is 3 to 1. So this one will be yellow, 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 and this one will be black. So 3 to 1 ratio. But again, they couldn't get this ratio, but the ratio they got is actually 2 to 1. Alright, so from here they suggested that the capital Y here are homozygote dominant actually die. Means when you do this crossing, so this is the yellow one, and you have, okay, so they got only yellow, 2 yellow, 1 black, so instead of 3 to 1. So what happened is actually the homozygote dominant embryo died, so the YY mice die, so the ratio will be only 2 to 1. Okay, so why this one will die? Okay, so actually this gene capital Y is called a lethal gene. It's actually killing the organism. So if we have 2 copies of this gene, it will be killing the organism. So when we are heterozygote, So the Y allele cause a yellow color of the coat in mice, and it is homozygote condition. The allele produce actually enough gene product to cause the mouse to die. So this gene actually is called, or this allele is called pleiotropic. So pleiotropic means it can affect many, it can have many functions in the same time. So pleiotropic, it will affect more than one phenotype. So here it can also cause, in case of heterozygote, it causes obesity, diabetes, tumor, and in homozygote, it will cause what we call this pre-implantation embryonic lethality. So it means it cannot, it doesn't, it can cause some problems. So the embryo cannot implant in the uterus, so the embryo will die. So this is what we call lethal gene. You have another example here in cats. So this is what we call Manx cat. They are tailless. Tailless cat with another trait is another trait that is caused by an allele, which is has dominant effect in heterozygote and is lethal in homozygote. So here the Manx is normal. Allele are denoted for M and small m for respectively for normal one and lethal one. So if you cross two tailless cats, what are you going to have? So M, capital M, small m, capital M, small m. What are you going to have? So yeah, the combination. So what are you going to have here? Instead of having 3 to 1, we will have 2 to 1. Why? Because this one will die. So homozygote dominant will die. So the heterozygotes will survive and will be tailless, will have no tail. Okay, but the small m, they will survive and they will have tail. Okay, so this is actually the example of little genes, and the ratio here will be 2 to 1. So 2 tailless and 1 with tail, because this one died off. Okay, now we move to a new parts, a new section is actually sex determination. In many animal species, chromosomes play a role in sex determination, and as you all know, we have two types of chromosomes. We have what we call autosomes and sex chromosomes. So the X and Y here shown in the picture are human chromosomes, sex chromosomes. So there are different types of this sex chromosomes. So in human, we have what we call XY system. In mammals, actually, XY system. Like in the case of human, we have 44 chromosomes and X and Y. If this one will make us a male, and if you have 44 chromosomes plus XX, then this one will be a female. Okay, so the Y chromosome here determines maleness, whether you are male or female. Drosophila also has the same system, Y and X and Y system. But birds they have another system. So you have what we call this ZW system. So when you are homozygote for the Z, for example, here it's a male, and heterozygote here is actually, or heterosome, it will be a female. So the sex chromosome are designed here by not X and Y but Z and W, so to distinguish them from X and Y in mammals. So the Y here will show the femaleness, and the Z will show the actually the femaleness. So and the female here, ZW, is called heterosome. Okay, and can be found also in fish and butterflies. Crickets and cockroaches, they have another system called XO system, XO or X0 system. So here in this case, if you have two copies of X, you are a female. If you have only one copy of X, you are a male. So that's why here you don't have another copy. You don't have an homologous chromosome for this one. So it's called X0, okay, or XO. All right. You have another system. This one we have already seen in parthenogenesis. This one is called the haploid-diploid system in bees. You know that in bees, the drones or male, they are haploid, the female, they are diploid. So the males are produced from eggs which are not fertilized, but the females, including the queen and the workers, they are produced from egg which is fertilized. So if the egg after the zygote is fed with honey, it will be a worker. If it is fed with royal jelly, it will be a queen. Okay, so here I just can you see in this picture here, this is actually are the chromosomes. This is are the autosomes from chromosome 1 to chromosome 22. They are autosomes. Of course, we have 2 copies of each chromosome, and here we have the sex chromosome. If you have XX is a female, XY is a male. So the sex chromosomes they are actually carry genes that control sexual development, and they also carry other genes that control other characteristics. And for example, here chromosome X imagine it contains up to 1,500 genes, and the Y chromosome contains 231 genes. But you can see the chromosome X is really a huge number of genes. Okay, so we are going to see some examples where the traits is actually caused by a gene located on the chromosome X. That's why we call it X-linked gene or traits. Means the gene is located on the chromosome X. These examples will include color blindness. Okay, some people who cannot see certain colors, and hemophilia A, where certain people lack the clotting factor for stop bleeding. So color blindness, or more accurately color vision deficiency, is an inherited condition that affects male more frequently than female. Alright, so control vision or color vision here is controlled by a gene located on the chromosome X, and this is explain why male are more affected than female, because male they have only one X, female they have two X. So male, if he has a X which is affected, means he will show the disease. The female has two X. If she receive one X which is affected, the other one will be normal, she doesn't show the disease. So here you have some numbers written here. So if you can see these numbers, means you don't have color blindness. So most of the cases of color blindness, 70 to 60% of them, they cannot see green color, and 30 to 40% of them, they cannot see red color. Okay, how the color blindness is inherited? We just said that the color vision actually actually the gene responsible for color vision is actually located on the chromosome X. So let's say the allele C for color is normal vision, and small c is the color blindness. So why we write normal allele is capital C? Because it's dominant over the other allele. And because it's located on the X, so we are going to write the X, and we are going to write the capital C here next to it. So we have to show that this is linked to X. This gene is located on chromosome X. And let's say here you have a mother who has capital X, X capital C, X small c, means she has one copy a normal gene, one copy of the normal gene, and one copy which is affected or mutated, and the father has a normal X. So if these two guys here marry, so what is the spring? So we will have, this is the gametes of the father, gametes of the mother. Then this girl here, she will receive normal genes for normal alleles from her mother and father, so she will be normal. This one here, she can receive normal X from her father, but affected one from her mother, so she will be normal. She can see colors, but she will be carrier. What does it mean? It means she can pass the disease to her kids. This boy here will receive the Y from his father and the X from his mother, the normal X. He will be normal. and this one will receive the Y from his father, but the mutated X from his mother. And this boy here will have color blindness. That's why you can see he's unhappy. So this boy here will have color blindness. So this was actually for the first example, color blindness. The second example is hemophilia A. So what is hemophilia A? So hemophilia, you can write it either way with the AE or E. So it's actually a condition where these people, they have a deficiency in clotting factor 8, which cause increased bleeding and usually affect males. Why? Because it's located on the chromosome X. So in the majority of cases, it is inherited as X-linked recessive trait. So it's recessive. So normal allele is dominant over the mutated allele. So normally when you cut yourself or you start bleeding, what will happen? In normal people, actually some clotting factors, including clotting factor 8, will cause a strong platelet plug to form, and fibrin network, if you want, will stop the bleeding. But in people affected with hemophilia A, they are lacking that factor 8, and this will cause the platelets to a weak reaction here, so the bleeding will continue. Okay, so here I just show you some animation. This is actually a cut. This one actually is covered in your chapter 10 in grade 10, chapter 9, sorry, in grade 10. So here you can see the cut in the blood vessel. So this is the red blood cells which are escaping from here. So this is the bleeding, and this white cell here is actually platelet. So what the platelet will do, the platelet will actually bind to this damaged part here, and it will start. We say that it will be activated. Alright, you can see here the shape change, and it will start releasing some chemicals to attract more platelets. So you can see here more platelets attracted, and they are releasing some chemicals here to attract more and more. So in this process here, what actually will happen is this we have what we call fibrinogen. This is a protein which is soluble in blood. When the platelets are activated, and including those clotting factors, this fibrinogen, which is soluble, will change into fibrin, which is insoluble, and it will form a network of fibers across the wound. So it will trap the red blood cells and form what we call a blood clot. It will stop the bleeding. Okay, so here you have another picture where more and more cells will join this wound to plug it. So this fibrinogen will make this fibrin. Okay, so you can see the network here, this net, it's trapping the RBCs, so the bleeding will stop. Okay, so okay, this factor 8 is actually is not alone, but you have many other factors. Okay, this one is not syllabus requirement, you don't have to memorize all this, but I just explain here what are those factors and how they are involved in bleeding, stopping bleeding. So here you have many other factors, factor 1, 2, 3, etc., etc. There, our factor 8 is here. This is factor 8. All right. So when you have any bleeding, so the platelet will be activated. So the platelets are activated, and they will start these reactions. And these reactions are involving here many factors. So we have factor 5, factor 7, factor 10, etc. So you can see our factor 8 is here, involved here. So all these reactions here involving all these factors to change fibrinogen into fibrin. So the fibrin will become insoluble and will make this network to trap the RBCs. So now if you are lacking this, means the reaction will stop here. Means this fibrinogen cannot form fibrin, means the bleeding will continue. So actually if you miss any of these factors, so nothing can happen, and the bleeding will continue. Okay, so all right, so now how is hemophilia inherited? Let's say we have here two normal parents, all right, and because this gene here is located on the X, we will write it X. We'll write the X. So let's say we annotate the H capital for normal allele, because the normal is dominant over the other allele for hemophilia. And here you have small h, and we have a carrier mother here with the normal husband. So what will happen? So what are they going to what will be the probability of their children to have the disease? So here this girl will receive normal X from both parents, so she will be normal. This one, she will receive one copy, normal copy from her father, and mutated copy from her mother. She will be carrier, means she can pass the disease to her children. And this boy received the Y from his father, the X here normal from his mother will be normal. But this boy, he received the Y from his father, but the mutated X from his mother, he will have hemophilia. He will be hemophiliac. Okay, this is a question for you to do. Anyway, so I will do here. You have actually here a normal mother, homozygote for normal, and this one you have a hemophiliac father. Let's see this boy here marry with someone who is normal. Okay, now does he have this hemophiliac boy or now a man? Can he pass the disease to his children? So what is the probability of having kids that will develop hemophilia? And what is the probability of female kids being carrier? So to answer these two questions, so it's very simple. You need to do a Punnett square. So this is the gametes of the father. This is the gametes of the mother. So what you can have here X normal H, here small h, means this one will be carrier, and here another one will be carrier, means these two girls, they receive the mutated X from their father. And the boys here, the boys inherit the Y from the father, and they will inherit the X from their mother. So the boys here will not have hemophilia. So to answer the first question, the probability of having kids that will develop hemophilia is 0. So you can see no one develop hemophilia here. So 0. The second question, what is the probability of having female kids being carrier? Actually, all the females will be carrier. Why? Because they inherit the X from their father, which is already mutated. Okay, so this is for hemophilia. Another example here, which is not sex linked, but it is influenced by sex. Okay, so inheritance of sex influenced characteristic. Example, baldness. So baldness is not caused by a gene located on chromosome X, but it is caused by a gene located on autosomes, but it is influenced by the sex, whether it's a female or a male. Okay, I explain more. So, baldness, this kind of traits, which are sex-influenced and sex-limited traits, they are not due to genes located on the chromosome X. They are not linked to chromosome X, but they are due to autosomal genes. So the expression of these genes is influenced by sex hormones. Example, in male you have testosterone, you have in female progesterone, estrogen. So both parents here, they will contribute equally to offspring. So example, pattern of baldness. It is actually sex-influenced trait. It is not caused by X gene or Y genes, but it is influenced by the sex. Let's say here the allele small or B, allele B and allele B plus. Let's say B is for bald, B plus is for normal hair, not bald. So if a person, so the male sex hormone means like testosterone, for example, it will play an important role in making the gene B for boldness dominant over the gene B plus in male with a genotype B plus. Okay, so here in male, if that person is a male B plus, what does it mean? His hormones or male hormones like testosterone, for example, is going to make this B dominant over the B plus, means the boldness dominant over the normal hair. So this male will be bald. So let's say now the B plus in a female. So because this female here, she doesn't have the male hormones, but instead of that she has estrogen and progesterone. So in this case, the B plus will be dominant over the B. So she will not be bald. Okay, so let's say that saying that. So you have genotype BB. You inherit both alleles are a small b or BB. Sorry, here for boldness. So the bold here, whether it's male or female, they will be bold. If you inherit B plus for normal hair, two alleles for normal hair, whether you are male or female, you will not be bold. But the only problem is when you are heterozygote. If you are B plus, will depend. Here, if you are a male, the B will be dominant over the B plus. So means it will be, the person will be bald. If you are a female, you don't have the male hormones, you don't have testosterone. So the B plus will be dominant over the B. So no, the female will not be bald. So here the expression of the gene B is influenced by the hormonal level, and only one copy of the baldness allele is sufficient to cause baldness in man. But you need two copies to cause baldness in women. Okay, so it behaves as a dominant in male because of the male hormones. Okay, and it behaves like a recessive in female. So that's half of the sons of a female carrier. If you have a female carrier, a female who is carrier, so what will happen? Half of her sons will be affected. So a heterozygote male will also pass the trait to half of his sons. Alright, so thus any traits that happen more frequently in male than female is suspected as either X-linked or sex-linked or sex-influenced. Okay, so you have an example here in how baldness is inherited. Let's say you have here a carrier mother, and this mother, of course, she is not bald. Why? Because she is a female. The B plus will be dominant over the B because she doesn't have the male hormones. Okay, and here the male is B plus means he doesn't have the allele for baldness, so he will have normal hair. This couple, are they? Is there any probability that they will have a child with baldness? To find out, we need to do the Punnett square. So this is the mother gametes, this is the father gametes, and here you will have B plus, and here B plus. Okay, so this one, whether it's male or female, they will not be bald. But this one, B plus, if it is a male, he will be bald. If it is a female, he will be not bald. Okay, so please do remember this one. If we got a BB for baldness, whether you are male or female, you will be bald. If you got B plus, whether you are male or female, you will not be bald. But if you are heterozygote, B plus means you got the allele for baldness and the allele for normal hair. This one, it will depend. It will depend whether you are male or female. If you are a male, means the B will be dominant over B plus and you will be bold. If you are a female, the B plus will be dominant over the B, so you will not be bold. And that's all. Thank you for your attention.